Hello, everyone, and thank you for being here. This presentation is going to cover some basic Winogradsky concepts and act as a primer for the Winogradsky and soil texture pre-lab and worksheet. First, we're going to do a bit of a review. We're going to discuss soil and biogeochemical cycling. And as you may recall, biogeochemical cycling is the relationship of life or bio. Geo meaning the earth, ground, or land. This would include the water, and which are specific atomic or molecular substances. When we put it all together, we get biogeochemical cycling, which describes the complex flows of matter through living organisms and the Earth's non living systems. Important to this is the concept of oxidation reduction reactions. This is going to come up time and time again, and we spend a lot of time front loading these concepts at the beginning of this course to get you very familiar with the terms, and it will get easier over time, I promise. Oxidation reduction reactions, for short, are called redox reactions. These are chemical reactions where one or more electrons are transferred between atoms or molecules. Here we can see in this example sodium, it's called Na in chemistry, and fluorine, which is called F in chemistry, abbreviated as such, um, exchange electrons. So sodium is not very electron greedy, and the electrons that it orbits its nucleus are easily transferable. Fluorine, on the other hand, is electron greedy. And so when these two elements become in close proximity with one another, the sodium is going to release one of its electrons, and fluorine is going to take one of those electrons. This is just an example. It's not something you will be tested on, but the purpose of this is to show the relationship of a redox reaction. One of these elements is being oxidized, and the other is being reduced. And we'll share in a moment what this looks like. But first, why are they called oxidation reduction reactions in the first place? Wouldn't it be easier just to call them electron exchange reactions? Well, let's talk about oxidation first. Why oxidation? It gets back to the study of oxidation reactions. And on Earth, oxygen is very prevalent, and it is one of the most electron greedy elements on Earth. And so in chemical reactions, it often is responsible for taking electrons, just like we saw fluorine in the previous slide, taking electrons from other atoms or molecules. The atom or molecule that loses an electron is then said to have been oxidized, and that's where the term oxidation comes from. So in the previous example, sodium was oxidized by fluorine. In other words, its electron was taken by fluorine. Oxidation. Why reduction? Well, this is related to the charge that electrons have. If you recall, electrons are negatively charged particles, and therefore, when an atom or molecule gains an electron, this means that the charge becomes more negative or it is reduced. So if the molecule had, let's say, a neutral charge, there was no positive or negative charge to it, and it takes an electron, it's going to gain an electron and become more negatively charged or more reduced. If we go back here, we can see that after the electron exchange occurs, sodium becomes positively charged as it's oxidized, and fluorine becomes negatively charged as it is reduced in its charge. Because the process of oxidation reduction occurs with electron movement, there's always going to be an oxidation coupled with a reduction. As we saw with the sodium fluoride reaction, there was the oxidation of sodium and the reduction of fluoride. And so one can't occur without the other. There's always a partnership in this dance. I want to lay down two rules of thumb that are going to add a lot of clarity for you when you're trying to figure out what's being oxidized and what's being reduced. Now, there is a lot of inorganic chemistry that doesn't involve these two atoms, but in all of our situations, it's going to be directly correlated to the amount of oxygen or hydrogen. So if you're looking at a compound in a chemical reaction and the number of oxygens in a molecule increases after the reaction, we would say that molecule is being oxidized. Makes sense. Uh, if the number of hydrogens is increasing in that molecule from before to after, then we would say that it is reduced. And so let's look at one of the examples we've shown before, and that is photosynthesis. The starting materials are CO2 and H2O, or carbon dioxide and water. And if we look at what the carbon turns into, it turns into glucose, or C6H12O6. And in carbon dioxide, we have one carbon for every two oxygens, CO2. But in glucose, we have one carbon for every oxygen. And so if we look at rule number one, the ratio of oxygens has declined. And so this molecule, as written above carbon dioxide, is reduced. Additionally, we can see that we added 12 hydrogens. So now there are, for every one carbon in glucose, there's two hydrogens. So fortunately for us, we see that rule number two applies to this equation too, giving us two clues that carbon dioxide is being reduced. If we look at water, we have two hydrogens to every one oxygen. And if we look at the other side of the equation, we see six O2s present. And so we've removed all of the hydrogen present. So hydrogen decreased. So if we look at rule number two, hydrogen didn't increase, it decreased. And so therefore it was oxidized. So look at the number of oxygens and hydrogens before and after, and that'll tell you a lot about if the molecule was reduced or oxidized. We can apply that same methodology to cellular respiration, which breaks down glucose with oxygen to create energy or kinetic energy, carbon dioxide, and water. Note that if we look at glucose, we have basically one carbon for every two hydrogens and one oxygen. And CO2 loses all of its hydrogen, and the number of oxygens is two for every one carbon. So if we look at rule number one, the ratio of oxygen increased. That would be an indication of oxidation. And rule number two, the ratio of hydrogen decreased. So instead of being reduced, we would assume here we have oxidation. So two clues that fit in nicely there. And if we look at oxygen in cellular respiration, we start with oxygen, we go to water. And so we've increased our hydrogen. That's a sign of reduction. And therefore, oxygen is reduced in this process. And there's our coupled reaction for both of those, a redox reaction for photosynthesis and a redox reaction for cellular respiration. If you can understand this concept, you can overlay that understanding for all of the other metabolic processes that we'll talk about in Chapter 2 and Chapter 3. Why are redox reactions important? Well, we're all quite familiar with energy being stored in batteries and transmitted through wires. And redox reactions, like what occurs in wires, are the movement of electrons, and that can release energy in cells in order to do work. So, this is going to be very important as we talk about environmental science further. So, we can imagine the chemical bonds as being the potential energy, like in a dam, the water that's stored above the dam or in the dam. And the electrons are that stored energy, potential energy. And we can release that energy by breaking apart those bonds and moving those electrons to a new molecule. There's two main reasons why organisms do redox reactions. One is to assimilate matter. So, take for example, plants that photosynthesize. They will do redox reactions to turn CO2 into organic matter in order to build the building blocks for cellular material so that they can grow and reproduce and do a variety of cellular functions. And to store or harvest that energy. So you might make glucose so that you can use it later. Humans, for example, will store energy in fat, which is a biomolecule that harnesses that energy and can be broken apart later to release that energy and use it to do work. One of the important molecules that we have to analyze in these redox reactions and in biogeochemical cycling is carbon. Carbon is the basis for all major biological molecules. So, whether it be fats, whether it be DNA, carbohydrates, et cetera, et cetera. Carbon has, as we can see, has 6 protons denoted in the upper right corner, and that's what makes carbon carbon. That's what defines carbon as being such. So now we have specific terms that are used to describe these different organisms based on how near or removed their energy gathering processes are from the ultimate energy source. And we can compare and contrast these two broad groups of organisms. We have autotrophs, and they produce their own energy through a mechanism chiefly called photosynthesis. Auto means self, and troph means to feed. So they're self feeding in this regard. That would include plants, algae, and cyanobacteria. On the other hand, we have heterotrophs. Hetero means other, so fed by others. So they consume the energy that is harvested by autotrophs in order to break apart those chemical bonds and serve their energy needs. These include all animals, all fungi, most bacteria, and small single celled organisms that are called protists. Let's examine the carbon cycle a little bit further and see how it relates to cellular respiration and photosynthesis. So, photoautotrophs, which we already discussed, acquire their energy from sunlight and feed themselves from CO2, absorb carbon dioxide from the atmosphere, and transfer it into biological molecules. We often show this biological molecule as glucose for simplicity's sake. These biological molecules can then be broken down in order to produce energy. This is usually through the process of cellular respiration, and it releases carbon dioxide into the atmosphere. Cellular respiration, if you notice, needs two starting materials. It needs our glucose or our biological matter, and it needs oxygen present for this aerobic process to occur. And then we can feed that carbon back into the system now that we've regenerated CO2. And you can see here that this is a cycle. So we need to have a balance between the amount of photosynthesis or cellular respiration, or we run up into a buildup of one of these components in this cycle. Now let's talk about carbohydrates a little bit more. Have you ever wondered why we call them carbohydrates and what carbohydrates are? Well, if you take a look at photosynthesis, it explains where the name is derived from. The term carbohydrates simply means carbon that has become hydrated with water. If you look at the chemical equation at the top, we have 6 carbon dioxides plus 6 water makes 1 glucose and 1 oxygen. And the chemical equation for all carbohydrates is going to be carbon plus water, and that makes CH2O. And we can confirm this by multiplying CH2O by 6. And that gives us, if you look on the right side, 6 carbons, 12 hydrogens, and 6 oxygens. And if we take a look at our glucose molecule in our equation above, we see that's exactly what we have. So in fact, glucose is simply hydrated carbon. Now, oftentimes we think that photoautotrophs or plants are kind of selfless organisms that just photosynthesize for the good of all of us heterotrophs. But that's really not the case. See, plants do photosynthesis to make biomolecules that serve them as well. And so they will make glucose or other biological molecules to build things around the cell. But also, when they need that kinetic energy available to them, they can go through the process of cellular respiration as well. As I described before, cellular respiration, one of the ingredients we've shown so far is oxygen there, and energy production can occur in the presence of oxygen. Obviously, we do that ourselves. Those are called oxic environments. This is the preferred area to degrade organic matter or do cellular respiration. Why? Because oxygen is so electron greedy. In the end, we can oxidize that organic matter with a high degree of efficiency, and that makes a lot of easy energy harvested. So we have a very efficient process. The issue is only the surface of the earth is abundant in free energy. But a lot of our environments are anoxic environments, meaning they lack oxygen or they're oxygen depleted. And so we can expect organic matter or elements to be reduced instead of oxygen. So oxygen not being present, we need to use something else to do the oxygen to oxidize our organic matter. So something else has to be, again, because it's a coupled reaction. Instead of oxygen being reduced, we need some other element or organic matter to be reduced. And that's what the Winogradsky lab is really going to exemplify. There's ways to do this, which we've discussed in our chapters. We can do fermentation. That's what makes, alcohol that we might consume or use in antiseptics. We might do lactic acid fermentation. That's what yogurts and cheeses and sauerkraut and pickling uses. Kimchi is another example. There's a form called anaerobic respiration, which is a little bit more oxidation than fermentation may do. Fermentation is a very partial form of oxidation of those organic compounds. And then methanogenesis takes it to an extreme. Methane is CH4. And so remember our rule number 2. There's a lot of hydrogen with that one carbon molecule. So it's basically the most reduced form of carbon as CH4. So that's methanogenesis. One of these examples might be nitrogen reduction. So we might not have oxygen available. Instead, we can use nitrogen. And so we can have nitrate or nitrate to nitrogen gas or N2. Or sulfur reduction is another. So if we can't reduce oxygen to make CO2, well, we'll reduce sulfur and we'll make CO4 or CO4 to hydrogen sulfide or sulfur. That's sulfur reduction. And we'll cover that in more detail in our Winogradsky lab. And so basically, we're replacing in our anoxic conditions, we're replacing oxygen with some other compound. Might be uh, NO3, it might be SO4. The point is, we're going to replace this with something else that can be reduced. And in this example, we replaced oxygen with nitrate or NO3, but we could do so with sulfur, sulfate, and instead of making NH4, we could make H2S or some other molecule like that that's reduced. Again, fermentation doesn't have those options. Instead, it just partially breaks down glucose. So in this example, lactic acid fermentation is a partial breakdown of glucose. And again, just to show you methanogenesis taking place here. So we can, in the presence of no biomolecules, organic carbon, we can take carbon dioxide and we can reduce it into methane. And then we also have methanotrophy that's part of this biogeochemical cycling where we turn methane in the presence of oxygen back into inorganic carbon. So methanogenesis, genesis being create, turns CO2 into CH4. That is clearly a reduction. We're losing oxygens in this process and gaining hydrogen. So very simple, clean cut. It means both rules of thumb for reduction. And that happens when, in the absence of oxygen. In the presence of oxygen, organisms love to consume methane. That's methane feeding or methanotrophy. And that's where we oxidize methane into CO2 again. And we can see this take place in the nitrogen cycle. I won't go into too much detail here, but at the top blue, that is in the presence of oxygen, we might oxidize our nitrogen compounds. And then in the absence of oxygen, we can reduce our nitrogen compounds. The sulfur cycle is going to be a lot more important in the Winogradsky lab. Sulfur is found naturally occurring in minerals and rocks, and through erosion, it gets released into soil and water. And once there, the sulfur gets converted into sulfate or it becomes oxidized. Or sulfur dioxide, which can be released through volcanic eruptions or combustion of fossil fuels. And we can see this cycle of sulfate being absorbed by plants and then consumed by heterotrophs. And then those wastes can go through a decomposition and create sulfate again. But what happens in these anaerobic environments or these anoxic environments? Well, we can use, without oxygen, organisms can, these are bacteria, can use oxygen to oxidize, or sorry, they can use sulfate to oxidize organic compounds. And that's what we see here with bacterial reduction of sulfate. Sulfate gets reduced into hydrogen sulfide or HS or H2S. And the reverse process of that, in the presence of oxygen, there can be bacterial oxidizers that oxidize sulfur. So it can go either way, but it really depends if oxygen is present or not. And that creates a cycle so we can recycle these chemicals, nutrients. And this is important to us as well because we use sulfate as fertilizer. Fertilizer sulfate needs to be the form that's taken up by plants. We also put a lot of sulfur into the atmosphere ourselves through industrial processes such as burning fossil fuels. All right, let's talk about Winogradsky um, culturing. And most culturing that you see in lab is done in two forms. It's done in a liquid culture or on a flat plate where the microbes are grown on top. And the two of these are not like what we see in the environment. In the environment, we have layers in which microbes grow throughout, as we saw in this previous slide. As we get deeper in the soil, we have a change in the availability of oxygen and a difference in the types of sulfur forms that are available. And these create homogeneous environments. The shaker shakes around and keeps all of the gas well mixed in the liquid. It doesn't provide a stratification that you might see in the ocean or a lake. And this doesn't have the layers that we see in sediment. That's where the Winogradsky column comes in. It's a lot different than any of those other laboratory cultures in that it creates a column. So we have a gradient that occurs here where we can study these different chemical reactions, metabolic processes, and the relationships between different organisms that do those different metabolic processes throughout that column. This column here is on display at the Harvard Museum of Natural History. Um, and it's a giant Winogradsky column. The purple light in part is from, or the purple coloring in part is from purple light that's shown on here to kind of exemplify it. So this allows, again, scientists to observe microbial interactions. And one of the important discoveries that was made here was chemoautotrophy. So far, we've talked about photoautotrophy that gets energy from sunlight in order to fix CO2 or feed themselves. Chemoautotrophy was discovered in the depths of these layers in a Winogradsky column. So, where sunlight might not be present, these organisms can fix their own carbon or harvest energy from chemicals in order to feed themselves or fix their own carbon into organic compounds. So, instead of photoautotrophy, this was named chemoautotrophy. And this is what Winogradsky discovered through his study of these columns. Here's an example of what these microbial mats look like. The coloration you're seeing here is due to different organisms capable of different metabolisms. On the surface, you often find photoautotrophs, um, and then we'll talk about this purple layer later on, and then you find chemoautotrophs down further in these layers. So they're using chemicals instead of sunlight to fix CO2. Chemicals such as hydrogen sulfide. Or, yes, if you've ever been to Yellowstone, you've seen in place Winogradsky column in nature. So these geothermal mats of Yellowstone are an example of such. The brown that you see present here on the edge is due to iron oxide vents, and the green down here is from green sulfur bacteria. Let's take that Winogradsky column and let's superimpose upon it a description of the different layers that are present in the column. Broadly speaking, a Winogradsky column can be broken down into three layers. The Let me get out my laser pointer here. We have the oxic zone, which we've discussed. This is where oxygen is readily present, and so it's going to be in the water overlay as well as the very top surface of the soil. Next, we have the suboxic zone. This is where oxygen is quite limited, but it's not a strongly reducing environment. There's still a lot of oxidized forms available. And then lastly, we have the anoxic zone. At the bottom here, this is where there's very little oxygen available, and it's a highly reduced environment is what we would call it. This is a highly oxidized environment. Okay. Now, depending on these different zones, you'll start to notice a trend here where we have different types of organisms that like to inhabit these different regions or niches. And those organisms are responsible for different forms of these elements existing. And in our Winogradsky column, we're going to be adding the element sulfur to this so that we can more easily exemplify the changes of sulfur. And what we'll see in a moment here is that that affects how much, say, you have in your sulfide. So our sulfide is going to be in the highest concentration down here at the bottom, where we have these sulfate reducing bacteria. So they reduce sulfate. Here's sulfate, and it's being reduced into sulfide. But as we go up the chain here, that sulfide is going to be consumed and all but gone here by the time we reach the surface in this oxic zone. So these Winogradsky columns, again, allow for observations of chemical cycling. We can see this in the left-hand column here of all of these chemical cycles taking place. Our columns, again, are supplemented with sulfur so that we can exemplify these different layers. And they're used to further our understanding of how elements are moved through Earth's biological systems. So again, chemoautotrophy, to recap, harnesses energy stored in chemicals. And in this example in the middle, in this yellow box, we can oxidize reduced sulfur right here, or maybe in other examples, methane or ammonia, which is NH4. And we can oxidize those to release energy, and we can harvest that energy to do work. This is very similar, as discussed, to how organisms will oxidize organic matter to harness energy, which is cellular respiration. And then this energy can be used to do whatever the cell wants, such as synthesize organic compounds from carbon dioxide or autotrophy. And that's where we get the name chemoautotrophy. As an example, we can look at colorless sulfur bacteria. They're called colorless because they lack any pigmentation, and they help recycle reduced sulfur. Here in the bottom here, we have our reduced sulfur in the presence of oxygen. So as this reduced sulfur gets mixed with upper layers, the colorless sulfur bacteria can take advantage of it. Okay, so they're going to recycle this reduced sulfur, and if it's reduced, they're going to oxidize it. And so they oxidize this reduced sulfur, and like cellular respiration does with organic carbon. So this reduced sulfur is replacing organic carbon. We're going to oxidize it in order to release energy from these reduced sulfur bonds, just like we release energy stored in reduced carbon bonds or glucose. The product of which, instead of CO2 as a waste, because we're not oxidizing glucose, is we're going to make sulfate as a waste product. We can see here. We can see that at the top here. We're going to be able to oxidize the reduced sulfur compounds, and then we can take that energy and put it to work. We can take CO2 and make it into a carbohydrate. Let's move along to soil texture. That's another component of the lab. And there are three different components of the solid phase of any soil. There is going to be the sand, the silt, and the clay, and they're defined by the type of size that exists in the soil. The largest particles are 50 to 200 micrometers in size. These are easily observable with the unaided eye. And so think about being at a beach. If you're at a beach, pretty much everything you see on that shoreline is going to be sand particles. So if you've ever held sand in your hand and rubbed it around and looked at it, you can see it's very grainy, like big chunks of kosher salt. Silt, on the other hand, is 2 micrometers to 50 micrometers. So it's a lot finer. You're not going to find it so much on the beach. You might find it um, in certain soils. So you can sort of see some texture to it. This is dry, but you can sort of, if when it's wet, you'll still be able to see a little bit of texture, a little bit of different colored particles in there that you can identify with your eye. I would describe, we describe this as kosher salt. Silt is more like finely ground pepper, where you can still see little pepper particles there. Or if you've ever brewed coffee before, finely ground coffee, like that you would use for espresso. So it's still a little grainy as you run it through your fingers. And when you're looking to identify clay in your soil column, these are the smallest particles, less than 2 micrometers. So this is as small as bacteria. You cannot see them with the eye here. This it looks clumpy because it's dried into little clumps here, but this is like if you were to wet clay, it'd be like a paste. And you wouldn't be able to see any individual particles like you could with pepper, right? Ground pepper. These three fractions influence the water flow through soil. Sand allows excellent drainage. And if you look at sand, imagine, if you had a bunch of balls that were very large balls and in a bag, the space between the balls where they don't quite marry flat is quite large. And so the water, as you water this soil, is going to slip through these cracks very easily. Silt is in between these smaller particles. They fit a little bit more tightly together. And so water is slowed down as it sticks to and tries to slide through these surfaces. So it has a moderate drainage and retention. And let me go back and say, because the water runs right through, this tends to wash off a lot of nutrients very easily. So it has poor nutrient retention in sandy soils. Silt is very moderate in both. Now, clay is very tightly packed together. There are little platelets here of molecules, and they stick together kind of like bricks. And if you imagine, once this gets compacted, you just can't really slide water through very easily at all. The water is going to stick to these clay particles. And so consequently, it has very poor drainage, and it's going to have good retention of nutrients. Soil texture is the combination of all of those blended size fractions together. So it's determined by the three of them. And a soil texture triangle has been created in order to try and define what types of textures we have. So you'll be able to use the soil texture triangle in your lab in order to determine what sort of soil is present for you. So here's an example of a soil texture test. And as I was describing before with sand, sand is really makes big clumps. So you can see individual particles here, and they're quite large. Let me get out my arrow here. So these quite large particles are present, big sand particles. The silt is much finer, like I said, kind of like fine coffee grounds or fine pepper, ground black pepper. You can still see this individual black particle, white particles, compounds throughout, but it's very fine and smooth like. And the clay is just like a paste that coats the top. And if you were to look very closely at the clay, you would not be able to discern any individual molecules here. Is just the silt protruding because it's settled unequally, but the clay level is very obviously a paste of sorts. So to use the triangle, we would look at our percent of each one of these. We can draw a line across our bottle and then measure the total height and say the total height was 10 inches. And let's say this first line was 6 inches from the bottom. We'd say 6 out of 10, that's 60% sand. And then from here to here is 3 inches. So we'd say that's 30% silt. And maybe the top is 1 inch, and that means it's 1 out of 10 or 10 or percent. So we would mark on the different sides here, our sand mark at 60%, our clay mark at 10%, and our silt mark at 30%. And we would draw a line across our 30%, straight across to land down here at the bottom side. Do the same with the sand. And we'd come across with our silt at 10%. And so we should be somewhere in the sandy loam region as the type of soil we have. There we go. So the tasks for you are first to complete the Winogradsky pre-lab. This is going to give you a little practice with those redox questions. And to make it easier, you'll see that each set of pre-lab questions have an assigned section from the textbook that you can use to reference to help you along the way. Then complete the Winogradsky worksheet. And the Winogradsky worksheet is going to be more based off of what you read in the Winogradsky lab protocol. So you're not to do the Winogradsky protocol this week. Don't worry about that. You'll do that next week. Right now, we're working on the Winogradsky pre-lab and the Winogradsky worksheet. Both are posted online for you to take. And then during next week, you're going to assemble the Winogradsky columns, and I will provide more information on that once we get there. And that's the end of this review. Please reach out to me if you have any questions at all. Thank you all for listening.